Thanks for coming out early, folks. One quick announcement. You've heard a little bit about the experimental lakes area and the changes in Canadian governance and the effects on this long-term program. You've heard about it more from an esoteric, like it's great for science, it's, you know, we need to keep this going, think of how the damage to our science. I just want to put a slightly personal spin on it. I think the only reason I stayed in aquatic science is because of the experimental lakes area. I didn't spend that much time there. But what it did is it exposed me to a way of doing work and an approach that profoundly changed what I wanted to do with my life. So the educational component of this is really important, and I don't think following this talk it could be downplayed. One of the things I think is important, though, is that we support the area, and I just, by way of an announcement, it's requested that if you would like to support the experimental lakes area, that booth 15 will be manned by David Schindler, Karen Kidd, Stu Hurwitz, and others. And for a $25 donation, you can help support the Institute, as well as get a cool t-shirt and literature on the interaction between population change and environmental degradation. Up until this point, we've honored and celebrated the sort of the career contributions of our colleagues. That is, some are short, more student-oriented, and some are more career-oriented. We're going to switch it around a little bit today, and we're going to talk about the fruits of our labor, the products of our science, and we're going to try and look at this from two different perspectives. One perspective is that of a young scientist. We come into this discipline, we want to change the world. That's why we're here. We think we can do it through the ASLO, and we want to produce something that has an effect on society and that we can use as our emblem. I did this. This is mine. On the other end, as we get further into our careers, we think about the cumulative effects of our research and our training. And we often look to products, at least I do, and I think I speak for some people, we try and think of how these products might have influenced our colleagues, our friends in society. So the two awards we're going to hear from right now are these two brackets. The first one, the Raymond Lindeman Award, is for a young career scientist for an outstanding paper produced in their first 10 or 12 years of their career. And then later we'll hear the Martin Award, which is more of a recognition of a paper that transformed our understanding. So with that in mind, today's winner of the Raymond Lindeman Award is Jillian Petersen. I'll just say a couple of brief words about it. As somebody who works in lakes, deep sea hydrothermal vents, that's a really cool alien environment, and Jillian's paper transformed how we understand these systems. Up until the time of the publication of the paper, the energy source for these bizarrely remote environments under 300 atmospheres pressure, 3 kilometers down in the middle of the ocean, was thought to be reduced sulfur compounds or methane. What Jillian showed was there's actually more going on there, and these vents can be very high sources of hydrogen, and this hydrogen can be a tremendous source of energy. But the thing that's interesting about her paper is it's not just that organisms can somehow magically capture hydrogen, it's that they're symbiotes with common genetic sequences, common evolutionary behavior that are embedded in all sorts of different organisms, crustaceans, mussels, etc., tube worms, and that they all can tap in on this energy source. In the nomination letter, it was pointed out that this paper has really transformed our understanding of these deep water energy sources, how such symbiotic relationships can be put together, and what their effect is on ocean ecology. With that, I'm going to invite a presentation by Jillian on her award, but it's going to be taped in. She's currently expecting her first child from Germany, and while she'd love to be here, she's sent her condolences and video messages.
Good morning, New Orleans. My name is Jillian Petersen. I'd like to welcome you to the Max Planck Institute for Marine Microbiology. And before I begin my presentation, I'd like to take this opportunity to thank firstly the ASLO President, Dr. John Downing, and the selection committee headed by Dr. Peter Levitt for giving me the opportunity to present my work to you today, and of course for the immense honor of being the recipient of this year's Lindeman Award. Today I will be telling you about how my collaborators and I discovered that hydrogen is an energy source for hydrothermal vent symbioses, And I will also give you a brief update on what's been happening since the paper came out in August 2011. Hydrothermal vents are unique ecosystems that exist in the total absence of sunlight. Here, the primary producers are chemosynthetic bacteria that use the chemicals emitted at vents as an energy source to power primary production. The abundant fauna at vents relies on symbiotic associations with such bacteria to provide for their nutrition. During my PhD studies, I had the unique opportunity to be involved in an interdisciplinary project funded by the German Research Foundation. This project aimed to investigate energy, material and life cycles at oceanic spreading axes. The Logachev hydrothermal vent on the Mid-Atlantic Ridge was one of the focal sites for this project. Logachev was chosen because it fascinated the geologists, the geochemists, the geophysicists, the oceanographers and the biologists equally. Because of its unique geological setting, Logachev vents are hosted in so-called ultramafic rocks. And because of reactions that happen in these rocks, the fluids at this site are particularly rich in hydrogen. In fact, the hydrogen concentrations at around 19 millimolar, measured in the pure hydrothermal fluids at Logachev, were at the time the highest hydrogen concentrations ever measured in nature. The biota at Logachev is dominated by mussels of the genus Bathymedilus, and the species is Bathymedilus pudeoserpentis. These Bathymedilus mussels host in their gills symbiotic bacteria that provide them with their nutrition. It was known that Bathymedilus from the Mid-Atlantic Ridge hosts a dual symbiosis. As you can see in cross-sections through the gill tissue, in pink are sulfur-oxidizing symbionts, the first type. Now these use reduced sulfur compounds as an energy source and CO2 as a carbon source. The second type is a methane oxidizer, shown here in green, which uses methane as an energy and carbon source. So since the original discovery of vents in 1977 until our paper in 2011, only two energy sources were proven to fuel hydrothermal vent symbioses. They were reduced sulfur compounds and methane. Our question was, given the high hydrogen concentrations at the Logachev vent field, can the mussel symbionts use it as an energy source? Our first approach was actually quite simple. We wanted to incubate the symbiont-containing gill tissues from Logachev to see if the hydrogen disappears. Or with hydrogen, to see if the hydrogen disappears. We did this on one of the first priority program cruises in 2005. We sampled the mussels from just over 3,000 meters depth. We brought them back on board, we dissected them, and we incubated them. Here you can see one of my collaborators, Frank Zielinski, who's busily dissecting a mussel for these incubations on board. We put the symbiont-containing gill tissue in closed vials, we added hydrogen, and over time we measured the hydrogen concentration in these vials. And our first results were electrifying. In our negative controls, that contained no symbionts, seawater, dead gill tissue and symbiont-free foot tissue, we saw very little hydrogen disappearance. In contrast, the hydrogen quickly disappeared in the incubations with the symbiont-containing gill tissue. To quote Nicole Dubillier, the head of the symbiosis group, PI on this project, at the time she said they just gobbled it up. So our journey to the cover of nature had begun. We spent the next years slowly gathering evidence for the use of hydrogen as an energy source by symbionts. We used a range of methods that took us from the symbiont genomes to the hydrothermal vent environment. And we used every precious minute of time on our almost annual research cruises during these years. In the first step, our incubations showed us that the gill tissues take up hydrogen and fix CO2 simultaneously. Therefore, hydrogen is used as an energy source to power primary production. In the next step, I was able to amplify and sequence the key gene for hydrogen uptake, HUP-L, from symbiont-containing gill tissues. We then used two novel methods to show that it's the sulfur-oxidizing symbiont, not the methane-oxidizing symbiont, that has and expresses the HUP-L gene. We used single-gene fluorescence in situ hybridization and immunohistochemistry combined with fish. Next, through sequencing of the symbiont genome, I obtained a large fragment of this genome that contained all of the genes for sulfur oxidation, CO2 fixation and hydrogen oxidation, more evidence that it's the sulfur oxidizer that's using the hydrogen. Finally, in collaboration with Pete Gergus from Harvard, we used an in situ mass spectrometer to show that hydrogen was actually disappearing in the mussel beds at Logachev. Our final question was, how widespread is hydrogen use in hydrothermal vent symbioses? The high hydrogen concentrations at Logachev are quite unique. Perhaps only the symbionts at this site are able to use hydrogen. However, as part of the priority program, we had the opportunity to visit another hydrothermal vent site called South Mar. This, unlike Logachev, is a basalt-hosted site, like most of the hydrothermal vents on Earth. And these don't have such unusually high hydrogen concentrations. Nevertheless, the symbionts in the mussels from these sites could also use hydrogen. Therefore, hydrogen use is not restricted to the ultramafic hosted sites with extremely high hydrogen concentrations. We then asked whether this ability might have been overlooked in the sulfur oxidizing symbionts of other vent animals. And indeed, I was able to find this HUP-L gene in the symbionts of the Rimicara shrimp from the Mid-Atlantic Ridge and also the symbionts from Riftia tube worms. This showed that hydrogen may in fact be powering many hydrothermal vent symbioses. But perhaps hydrogen use was not just overlooked in symbioses. I'd now like to finish up telling you about our study and give you a brief overview about what we and others have found out since then. First, I would like to tell you a bit more about the sulfur and hydrogen oxidizing symbionts of Bathymedilus mussels. According to the 16S ribosomal RNA gene, a universal genetic marker for bacteria, the sulfur oxidizing symbionts of mussels are closely related to sulfur oxidizing symbionts of vesicomyid clams from the deep sea. But they're actually even more closely related to a very widespread pelagic group of bacteria, the SUPO5 sulfur oxidizers. Now, at the time of our study, no hydrogenase genes had been found in SUPO5 or in the clam symbionts, even though genomes and metagenomes were available for these. We therefore hypothesized that the Bathymedilus symbionts had acquired the ability to use hydrogen through horizontal gene transfer of the hydrogenase genes. Recently, free-living SUP5 bacteria were discovered abundant in a hydrothermal plume associated with the Guaymas Basin hydrothermal vents. The hydrothermal plume is an area of mixing in the water column between pure hydrothermal fluids and deep sea water. As I said before, no SUP5 bacteria able to use hydrogen had previously been found. But amazingly, a recent study from Greg Dick's lab at the University of Michigan, which is currently online early at PNAS, showed that these hydrothermal vent plume SUP5s actually have hydrogenase genes and they express them. When we look at the phylogeny of the HUP-L gene, we can see that the hydrogenases of Bathymedilus symbionts and Guaymas SUP5 are very closely related, and this reflects their very close evolutionary relationship based on the 16S ribosomal RNA gene. However, these do not group together with hydrogenase genes from other sulfur-oxidizing gamma proteobacteria, such as the symbiont of Riftia tube worms or Allochromatium or Thiocapsa. Instead, they group together with hydrogenases from alpha and beta proteobacteria. which are far more distantly related. The most striking thing about these results is actually the fact that the hydrogenases from these Rimicaris symbionts are closely related to the other vent organisms, the Bathymedilus mussel symbionts and the SUP5s. Horizontal gene, this is unexpected because these Rimicaris symbionts and SUP5s and the Bathymedilus symbionts are not closely related. So horizontal gene transfer does seem to have played a role in the evolution of hydrogen use by vent symbionts. Even more intriguingly, I've recently been involved in a metagenome study of these Rimicaris shrimp symbionts, which showed that the symbiont actually has two hydrogenase genes. We found the one that was closely related, the one that we'd previously found, closely related to the Bathymedilus symbionts and SUP5. The second was actually very closely related to the hydrogenase from the Riftia symbionts. Riftia and Rimicaris are so far not known to co-occur physically at one vent site. So we think, could this be an example of habitat-specific horizontal gene transfer? Similar habitat-specific gene transfer events have recently been shown for human gut microbes. So a team around Eric Alm at MIT recently showed that horizontal gene transfer, which is a powerful force in microbial evolution, is shaped primarily by ecology rather than phylogeny or geography. They showed that recently transferred genes between bacteria occupying a well-defined niche are especially likely to reflect adaptation to that niche. So coming back to the hydrothermal vent environment, hydrogen oxidation seems to be one of the key adaptations of life to hydrothermal vents by both symbiotic and free-living bacteria. Looking into how this adaptation evolved will be one of the things that we plan to do in the future that you should look out for. I would like to conclude by thanking the team that was involved in this paper. In particular, I thank Frank Zielinski, with whom I gladly share first authorship of this paper, and to Nicole Dubillier, head of the Symbiosis Group at the Max Planck Institute in Bremen, and the principal investigator on the study. Finally, I wish I could have joined you today in person, but since this was not possible, I'd like to invite you to visit me at my website or send me an email if you have any questions. Thank you for your attention, and I wish you a successful and productive ASLO 2013.